This session is in continuation with our previous class on the reactions of biological system, lecture number 1, where we discussed biological reactions from biophysical and thermodynamic point of view. Today, in this class, we are going to discuss the reactions of the biological system from biochemical point of view with special reference to the enzymes, the landmark biological catalysts. The second part of our talk will focus on the specialized reactions of the biological system, namely photosynthesis, replication, transcription, and translation. The biological reactions are no different from the chemical reactions, but they take place in controlled biological environments under the supervision of the enzymes. It is this regulation that makes the reactions of the biological systems so exclusive and exquisite. Control takes place in the biological reactions with respect to time and place. For example, reactions like transportation of molecules and signal transduction reactions take place associated with the membrane, whereas histone acetylation and deacetylation type of reactions take place associated with the nucleoid in prokaryotes, and in the case of eukaryotes, it happens inside the nucleus. In the eukaryotic system, where there is compartmentalization, reactions take place in specialized compartments. For example, cytochrome oxidase reactions always take place inside mitochondria, whereas acid hydrolases work inside the lysosomes. Biological reactions are also precise with respect to time. In prokaryote, where the cell cycle is not so well defined, still there are reactions that take place immediately after the cell cycle, while some other reactions take place just before the cell cycles. This phenomena is even clear in eukaryotes, where the cell cycle is discretely divided into G1, S, G2 and M phase. Reactions like replication takes place strictly in S phase only. It is important to know about the large molecules that provide cellular informations. DNA and RNA are such large informational macromolecules. DNA can be called strictly as an informational macromolecule because till date no function has been found to be associated with DNA. DNA serves as the genetic material for most of the organisms on the earth. For example, E. coli contains 4.2 times 10 to the power 6 base pair DNA, or 42 lakh base pair of DNA, which is the storehouse of information for all the biological reactions that take place inside the E. coli cell. The human genome is of the size of 3 times 10 to the power 9 base pair. RNA, on the other hand, serves as the storehouse of information for many viruses, for example, TMV. RNA is also produced as the first step of gene expression through transcription. RNA is also an informational macromolecule, but enzymatic functions has also been ascribed to RNA. Peptidyl transferase activity, RNase P activity are such examples. Weak interactions for structural stability and alteration is of extreme importance to understand the reactions of biological system. Weak interactions are important because they are easy to be made and broken, and enzymes deploy these weak interactions to convert substrate into products in the reactions of the biological system. Weak interactions that are predominant in biological systems include hydrogen bonds, hydrophobic interactions, and van der Waals interactions. As we see that all naturally occurring DNA are double-stranded. These double-stranded DNA are held in double-stranded configuration by nothing else but hydrogen bonds. 2 hydrogen bonds between adenine and thymine and 3 hydrogen bonds between guanine and cytosine held the 2 strands of DNA in double-stranded configuration. Hydrophobic interactions are of importance for holding the biological membranes. Most of the biological membranes have a lipid bilayer type of a structure where the bilayer of lipid is held together by hydrophobic interactions between the nonpolar tails of the lipids in polar aqueous environment. Van der Waals interactions are of extreme importance because they hold the 3-dimensional structures of the proteins. Most of the biological reactions go in a downhill fashion, meaning the energy of the reactants for biological reactions are most of the times more than that of the products, or in other words, for most of the biological reactions, the delta G value or the Gibbs free energy change is negative. There are few exceptions in the case of anabolic reactions where the energy of the product is higher than those of the reactants. It is to be noted that those reactions can only happen in nature when they are coupled with some other reactions where there is even greater fall of the delta G value. The thousands of reactions that happen in the biological system can really be grouped into few classes. For example, the oxidation reduction reactions, the reactions that cause movement of functional groups within and between molecules, the reactions that are involved in addition or removal of water, or the reactions that are involved in making and breaking of bonds. All these reactions are actually catalyzed by the enzymes, and the enzyme action is very simply depicted in this cartoon, where the enzyme is shown in green. It binds to a substrate shown in red, and the enzyme substrate complex is formed, which is eventually catalyzed to form the enzyme product complex. The enzyme product complex dissociates to form the product and the enzyme. So, the final product of the reaction is that the substrate got converted to the product. However, the enzyme remains intact. It can once again bind to another substrate molecule to convert it to product. It is to be noted that enzymes cannot alter the energy level of reactants and products of an enzyme catalyzed reaction, or in other words, enzymes cannot alter the delta G value of any reaction. All they can do is that they can lower the activation energy. For most of the biological reactions, there is an energy barrier as shown in this diagram with the blue curve. When this reaction is catalyzed by enzymes, enzyme lowers the energy of activation as shown in the red curve. So, enzyme facilitates formation of more product from the reactants. Now, all the reactions that enzyme participate in, depending on the type of reaction they catalyze, enzymes can be classified into 6 groups. Number 1 are the oxidoreductases. They catalyze transfer of hydrogen or oxygen atoms or electrons from one substrate to the other. They are called dehydrogenases or oxygenases, and examples include lactate dehydrogenase, succinate dehydrogenase, or cytochrome oxidases. The second group are the transferases, which transfer a specific group like a phosphate group, a methyl group, or an amino group from one substrate to the other. They are named as the kinases or transaminases. T4 polynucleotide kinase, acyl transferases are examples of such group. The third group includes the hydrolases, which hydrolyze a substrate, and digestive enzymes are best examples of this class. Isomerases change molecular forms of a substrate, meaning they change groups within the substrate. So, the overall formula of the substrate remains the same. Epimerases, isomerases are examples of this class. The fifth class of enzymes are the lyases, which cause non-hydrolytic removal of a group or addition of a group to the substrate. The last group is the ligase, which are responsible for joining two molecules by forming new bonds. Citric acid synthetases, T4-DNA ligases are the examples of this class. Now, in my previous slide, I stated that reactions of the biological system can be put together into 4 different types. However, they are catalyzed by 6 different classes of enzymes. This slide bridges up the reactions that are catalyzed by the enzymes. For example, oxidation reduction reactions are catalyzed by oxidoreductases. Movement of functional groups within and between molecules are catalyzed by group number 2 and 4, namely the transferases and the isomerases. Addition and removal of water are mediated by group number 3 enzymes, the hydrolases, and last, the making and breaking of bonds are catalyzed by group number 5 and 6, namely the lyases and the ligases. Controlling the biological reactions is of extreme importance, and in the biological system, control can operate at many different levels. For example, enzyme action can be regulated by or at the gene expression level. As we can see from this scheme, most of the enzymes are proteins and they are transcribed and translated from a gene. Now, a gene is first transcribed into a primary transcript and it undergoes processing of the RNA to form an mRNA. In eukaryotic system, it is transported then from the nucleus to the cytoplasm where it is translated. So, control in expression can occur at all these different levels. For example, there can be transcription control where the first step of gene expression, that is the transcription, is controlled. Then, after the primary transcript is made, control can act on processing the RNA to form the mRNA. In eukaryotic system, transport of RNA from the nucleus to the cytoplasm can also be controlled, and finally, the RNA can be controlled at the translational level to form the protein. Now, in eukaryotic system, controls can operate in all these 4 levels. In prokaryotic system, however, most of the control lies at the transcription level, that is at the very first step where a gene is transcribed into an mRNA. Regulation of enzyme can also take place through allosteric modulation. As shown in this scheme, an enzyme can have 2 subunits. The catalytic subunit where the substrate binds to be converted to the product, and there can be an allosteric subunit where allosteric modulators like allosteric activators or allosteric inhibitors can bind. As shown in this cartoon, there can be enzymes that can be controlled by allosteric inhibition. When the allosteric inhibitor binds to the allosteric site, it causes a change in the conformation of the enzyme molecule, so the substrate cannot bind to the enzyme anymore and the reaction doesn't take place. This is an example of allosteric inhibition. Similarly, there can be allosteric activation, meaning the substrate can bind to the enzyme only when at the allosteric site the allosteric activator is bound. Binding of the allosteric activator changes the conformation of the enzyme in such a way that it facilitates binding of the substrate to the enzyme and the substrate is converted to the product. This is called allosteric activation. At this stage, it has to be noted that not all enzymes are allosterically controlled. There are enzymes which doesn't have any allosteric subunit, whereas there are enzymes which are allosterically inhibited, there are enzymes which are allosterically activated, and there are examples of enzymes which are both allosterically activated as well as allosterically inhibited. Let us go through some of the examples. For example, the glycogen synthase, also called the UDP glucose glycogen glycosyl transferase, is the enzyme that joins up glucose units to form glycogen. Now this glycogen synthase can be phosphorylated by glycogen synthase kinase 3, also called the GSK3. Now this GSK3 can phosphorylate the glycogen synthase, and this phosphorylation leads to inactivation of the enzyme. It can no more convert glucose to glycogen. So in the case of glycogen synthase, phosphorylation leads to inactivation of the enzyme. So phosphorylation is an important reaction of the biological system. There are examples where phosphorylation inactivates an enzyme. There are examples as well where phosphorylation can activate an enzyme. Activation and deactivation by phosphorylation is found in abundant amount for signal transduction and metabolic pathways. As seen in the cholesterol biosynthetic pathway, the major regulatory enzyme HMG-CoA reductase is inhibited by phosphorylation, whereas two other enzymes that are key regulatory enzymes in the utilization of cholesterol, namely the acyl-CoA cholesterol O-acyl transferases or ACATases with EC number 2.3.1.26 and cholesterol 7 alpha hydroxylase with EC number 1.14.13.7. These two enzymes are activated by phosphorylation. Enzyme action can also be regulated by controlling the environment in which they catalyze the reactions. This example shows that the digestive enzyme pepsin has a pH optima at around 2.5. It is active in the stomach where the pH is about 2. Now, when the food along with pepsin moves into the intestine, bile juice comes and gets added and the pH rises up to 8 and pepsin gets deactivated. Now trypsin, another protein digesting enzyme secreted from the pancreas, gets activated because trypsin has a pH optima of around 8. So these examples are pH dependent inhibition of pepsin in small intestine is an example where a reaction is controlled by the change of the environment. Finally, enzymatic reactions can also be controlled by feedback inhibition. These type of examples are found in metabolic pathways. As shown in this scheme, a substrate A gets converted to G in series of reactions where 6 enzymes are involved. Like A is converted to B, say by enzyme E1, B is converted to C by enzyme E2, and so on. Now it has been observed in many biochemical pathways that the end product of the reaction, that is in this case the product G, inhibit one or the other enzymes that are located upstream to these reactions. For example, here we see that the product G inhibits the enzyme that converts C to D. This is how many metabolic reactions are turned off when sufficient amount of end products are formed. This type of controls are called feedback allosteric modulation. Now we reach the second part of our today's talk, where we will discuss about the specialized reactions of the biological system, namely photosynthesis, replication, transcription, and translation. These are specialized reactions found only in the biological system. These are very elaborate reactions and lots of enzymes, cofactors and other molecules take part in an orchestrated fashion to form a product. Photosynthesis is the most important biological process where the energy of the sunlight is trapped into chemical energy by photosynthetic plants and other microorganisms. This is the only reaction that supports the entire aerobic life on Earth. The photosynthetic organisms are called photoautotrophs because they can prepare their own food. The higher organisms depend directly or indirectly on these photoautotrophs for their food. The overall reaction of photosynthesis can be written as 6 molecules of carbon dioxide taken from atmosphere gets joined with 6 water molecules in presence of sunlight to form the sugar C6H12O6, and 6 oxygen molecules are released in the environment. Replication is the important reaction that is responsible for maintenance of the genetic inheritance. In the biological system, when a cell reproduces, it has to ensure that its offsprings or the 2 daughter cells get similar amount of DNA as that of the mother cell. Replication ensures that. As in this diagram, the old strands or the template strands are shown in blue, the new strands are shown in red, and the DNA polymerases are shown in green. In the leading strand, the DNA is synthesized in a continuous fashion, whereas in the lagging strand, DNAs are synthesized in a fragmented way as Okazaki fragments. The overall reaction of replication can be shown as the formation of phosphodiester bonds, where the hydroxyl group of C3 attacks the alpha phosphate group and a 5' to 3' phosphodiester linkage is created between the deoxyribonucleotides. Transcription is the next important reaction in the biological system. It happens almost in all living cells, and transcription is the first step of gene expression. Transcription takes place on the surface of RNA polymerase. As shown in this diagram, the double-stranded DNA is opened up in the surface of RNA polymerase, and the new RNA strand that is synthesized is shown in blue. The strand against which the RNA is synthesized is called the template strand, while the strand complementary to the template strand is called the coding strand. The overall reaction of transcription is the formation of phosphodiester bond between ribonucleotides, exactly the same way phosphodiester bonds are formed between deoxyribonucleotides in replication. Translation is another important reaction of the biological system that happens in almost all cells. This is the final step of gene expression. On the ribosome surface, amino acyl tRNA molecules called the translational adapters bring the amino acids, and codon-anticodon interaction take place and peptide bond is formed. The overall reaction as shown in this figure is when 2 amino acids get joined together by peptide bond formed between OH group of the carboxylic acid group of one amino acid and the hydrogen of the amino group of the other amino acid. So CONH are the main players of the peptide bond. Informational macromolecules like DNA and RNA holds the remote control for the reactions that happen in the biological system. Weak interactions are instrumental for the biological systems because they are easy to be made and broken. Most of the biological reactions are downhill in terms of thermodynamics because the gives free energy change at the delta G value is negative. Enzymes work by lowering the activation energy and they facilitate formation of more products from the reactants. Control is the hallmark of the reactions that happen in biological system. Since most of the reactions of the biological system are catalyzed by enzymes, controlling the enzymes confer control on the reactions of the biological system. And enzymes can be controlled in many different levels, like the expression level, by allosteric modulation, by feedback inhibition, and by controlling the environment of the reaction of the enzymes. Specialized biological reactions like photosynthesis is very important because it can trap the energy of sunlight into the energy of chemical bonds. Photosynthesis is the vital process that supports all aerobic life on Earth because photosynthesis adds oxygen to the atmosphere. Photosynthetic organisms are called photoautotrophs because they prepare their own food, and heterotrophs like the human beings directly or indirectly rely on these autotrophs for their food. Replication is the fundamental reaction of the biological system that is responsible for genetic inheritance, and replication ensures that after cell division, the two daughter cells get similar DNA as that of their mother. Transcription is the first step of gene expression, and it is found in almost all organisms, and transcription is the step where all the controls in prokaryotes are observed. Translation is the final step of gene expression, and translation is the predominant of all the processes that always take place inside the cell.